Proteasomes. As you have just learned, lysosomes degrade proteins delivered to them in vesicles. Cytosolic proteins also require disposal at certain times in the life of a cell. Continuous destruction of unneeded, damaged, or faulty proteins is the function of tiny barrel-shaped structures consisting of 4 stacked rings of proteins around a central core called proteasomes. For example, proteins that are part of the metabolic pathways are degraded after they have accomplished their function. Such protein destruction plays a part in negative feedback by halting a pathway once the appropriate response has been achieved. A typical body cell contains many thousands of proteasomes in both the cytosol and the nucleus. Discovered only recently because they are far too small to discern under the light microscope and do not show up well in electron micrographs, proteasomes was so named because they contain a myriad of protease enzymes that cut proteins into small peptides. Once the enzymes of a proteasome have chopped up a protein into smaller chunks, other enzymes that break down the peptides into amino acids, which can be recycled into new proteins. To repeat that, discovered only recently because they are far too small to discern under light microscope and do not show up well in electron micrographs, proteasomes was so named because they contain myriad proteases, enzymes that cut proteins into small peptides. Once the enzymes of a proteasome have chopped up a protein into smaller chunks, other enzymes then break down the peptides into amino acids, which can be recycled into new proteins. Some diseases could result from failure of proteasomes to degrade abnormal proteins. For example, clumps of misfolded proteins accumulate in brain cells of people with Parkinson's disease and Alzheimer's disease. Discovering why the proteasomes fail to clear these abnormal proteins is a goal of ongoing research. Mitochondria. Because they generate most of the ATP through aerobic respiration, mitochondria are referred to as the powerhouses of the cell. A cell may have as few as 100 or as many as several thousand mitochondria. Depending upon the activity of the cell, active cells, such as those found in the muscles, liver, and kidneys, which use ATP at a high rate, have a large number of mitochondria. For example, liver cells have about 1,700 mitochondria. Mitochondria are usually located within the cell where oxygen enters the cell or where the ATP is used, for example, among the contractile proteins in muscle cells. A mitochondrion consists of an outer mitochondrial membrane and an inner mitochondrial membrane with a small fluid-filled space between them. Both membranes are similar in structure to the plasma membrane. The inner mitochondrial membrane contains a series of folds called cristae. The central fluid-filled cavity of a mitochondrion, enclosed by the inner mitochondrial membrane, is the matrix. The elaborate folds of the cristae provide an enormous surface area for the chemical reactions that are part of the aerobic phase of cellular respiration. The reactions that produce most of the cell's ATP. The enzymes that catalyze these reactions are located on the cristae and in the matrix of the mitochondria. Like peroxisomes, mitochondria self-replicate, a process that occurs during times of increased cellular energy demand or before cell division. Synthesis of some of the proteins needed for mitochondrial function occur in the ribosomes that are present in mitochondrial matrix. Mitochondria even have their own DNA in the form of multiple copies of a circular DNA molecule that contains 37 genes. These mitochondrial genes control the synthesis of 2 ribosomal RNAs, 22 transfer RNAs, and 13 proteins that build mitochondrial components. Although the nucleus of each somatic cell contains genes from both your mother and your father, mitochondrial genes are inherited only from your mother. The head of a sperm, the part that penetrates and fertilizes an oocyte, normally lacks most organelle, such as mitochondria, ribosomes, endoplasmic reticulum, and the Golgi complex, and any sperm mitochondria that do enter the oocyte are soon destroyed. Nucleus. The nucleus is a spherical or oval-shaped structure that usually is the most prominent feature of a cell. Most cells have a single nucleus, although some, such as mature red blood cells, have none. In contrast, skeletal muscle cells and a few other types of cells have multiple nuclei. A double membrane called the nuclear envelope separates the nucleus from the cytoplasm. Both layers of the nuclear envelope are lipid bilayers, similar to the plasma membrane. The outer membrane of the nuclear envelope is continuous with rough endoplasmic reticulum and resembles it in structure. Many openings, called nuclear pores, extend through the nuclear envelope. Each nuclear pore consists of a circular arrangement of proteins surrounding a large central opening that is about 10 times wider than the pore of a channel protein in the plasma membrane. Nuclear pores control the movement of substances between the nucleus and the cytoplasm. Small molecules and ions move through the pores passively by diffusion. Most large molecules, such as RNA and proteins, cannot pass through the nuclear pores by diffusion. Instead, their passage involves an active transport process in which the molecules are recognized and selectively transported through the nuclear pore into or out of the nucleus. For example, proteins needed for nuclear function move from the cystole into the nucleus. Newly formed RNA molecules move from the nucleus into the cystole in this manner. Inside the nucleus are one or more spherical bodies containing nucleoli. They contain nucleoli that function in producing ribosomes. Each nucleolus is simply a cluster of protein, DNA, and RNA, and is not enclosed by a membrane. Nucleoli are the sites of synthesis of rRNA and assembly of rRNA and proteins into ribosomal subunits. Nucleoli are quite prominent in cells that synthesize large amounts of proteins, such as muscle and liver cells. Nucleoli disperse and disappear during cell division and recognize once new cells are formed. Within the nucleus are most of the cell's hereditary units, called genes, which control cellular structure and direct cellular activities. Genes are arranged along chromosomes. Human somatic cells have 46 chromosomes, 23 inherited from each parent. Each chromosome is a long molecule of DNA that is coiled together with several proteins. This complex of DNA proteins and some RNA is called chromatin. The total genetic information carried in a cell or an organism is its genome. In cells that are not dividing, the chromatin appear as a diffuse granular mass. Electron micrographs reveal that chromatin has a beads on a string structure. Each bead is a nucleosome and consists of double-stranded DNA wrapped twice around a core of 8 proteins called histones, which help organize the coiling and folding of DNA. The string between the beads is linker DNA, which holds adjacent nucleosomes together. In cells that are not dividing, another histone promotes coiling of nucleosomes into a larger diameter chromatin fiber, which then folds into large loops. Just before cell division takes place, however, the DNA duplicates, and the loops condense even more, forming a pair of chromatids. As you will see shortly, during cell division, a pair of chromatids constitutes a chromosome, the main part of a cell. Their descriptions and their functions are summarized as follows. Plasma membrane, fluid mosaic lipid bilayer, phospholipids, cholesterol, and glycolipids, studded with a protein, surrounds cytoplasm. Its function is that it protects cellular contents, makes contact with other cells, contains channels, transporters, receptors, enzymes, cell identity markers, and linker proteins, mediates the entry and exit of substances. Cytoplasm, cellular contents between the plasma membranes and nucleus, systole and organelle. Sites of all intracellular activities except those occurring in the nucleus. Systole, composed of water, solutes, suspended particles, lipid droplets, and glycogen granules. Medium in which many of cells' metabolic reactions occur. Organelle, specialized structures with characteristic shapes. Each organelle has specific functions. Cytoskeleton, network of 3 types of protein filaments, microfilaments, intermediate filaments, and microtubules. Maintain the shape and general organization of cell contents responsible for cellular movement. Centrosome, a pair of centrioles plus pericentriolar material. This pericentriolar material contains tubulins, which are used for growth of the mitotic spindle and microtubule formations. Cilia and flagella they are motile cell surface projections that contain 20 microtubules in a basal body. Cilia move fluids over a cell's surface. Flagella move an entire cell. Ribosome, composed of 2 subunits containing ribosomal RNA and proteins, may be free in systole or attached to rough endoplasmic reticulum. Their function is protein synthesis. Endoplasmic reticulum is a membranous network of flattened sacs or tubules. Rough ER is covered by ribosomes and is attached to the nuclear envelope. Smooth endoplasmic reticulum lacks ribosomes. Rough endoplasmic reticulum synthesizes glycoproteins and phospholipids that are transferred to cellular organelle, inserted into the plasma membrane, or secreted during exocytosis. Smooth endoplasmic reticulum synthesizes fatty acids and steroids, inactivates and detoxifies drugs, removes the phosphate group from glucose 6-phosphate, and stores and releases calcium ions into muscle cells. The Golgi complex consists of 3 to 20 flattened membranous sacs called cisternae, structurally and functionally divided into entry face, medial cisternae, and exit face. Entry, CIS face, accepts proteins from rough endoplasmic reticulum. Medial cisternae from glycoproteins, glycolipids, and lipoproteins. Exit, transface, modifies the molecules further, then sorts and packages them for transport to their destinations. Lysosome, vesicles formed from Golgi complex, contains digestive enzymes, fuses with and digests contents of endosomes, pinocyte vesicles, and phagosomes, and transport final products of digestion into systole. Digest worn-out organelle, autophagy, entire cells, autolysis, and extracellular material. Peroxisomes, vesicle containing oxidase, oxidative enzymes, and catalase, decompose hydrogen peroxide. New peroxisomes bud from pre-existing ones. Oxidizes amino acids and fatty acids, detoxifies harmful substances such as alcohol, produces hydrogen peroxide. Proteasome, tiny barrel-shaped structures that contain protease, proteolytic enzymes, degrades unneeded, damaged, or faulty proteins by cutting them into small peptides. Mitochondrion consists of an outer and an inner mitochondrial membrane, cystae, and matrix. New mitochondria form from pre-existing ones. The site of aerobic cellular respiration reactions that produce most of a cell's ATP. The nucleus consists of a nuclear envelope with pores, nucleoli, and chromosomes, which exit as a tangled mass of chromatin in interphase cells. Nuclear pores control the movement of substances between the nucleus and the cytoplasm. Nucleoli produce ribosomes, and chromosomes consist of genes that control cellular structures and direct cellular function. Protein synthesis. The object of this section is to describe the sequence of events in protein synthesis. Although cells synthesize many chemicals to maintain homeostasis, much of the cellular machinery is devoted to synthesizing large numbers of diverse proteins. The proteins in turn determine the physical and chemical characteristics of cells and therefore of the organisms formed from them. Some proteins help assemble cellular structures such as plasma membranes, the cytoskeletons, and other organelles. Others serve as hormones, antibodies, and contractile elements in muscular tissue. Still others act as enzymes regulating the rates of the numerous chemical reactions that occur in cells, or transporters carrying various materials in blood. Just as genome means all of the genes in an organism, proteome refers to all of an organ's proteins. In the process called gene expression, a gene's DNA is used as a template for synthesis of a specific protein. First, in a process aptly named transcription, the information encoded in a specific region of DNA is transcribed to produce a specific molecule of RNA. In a second process, referred to as translation, the RNA attaches to a ribosome where the information contained in the RNA is translated into a corresponding sequence of amino acids to form a new protein molecule. DNA and RNA store genetic information as sets of 3 nucleotides. A sequence of 3 such nucleotides in RNA is called a base triplet. Each DNA base triplet is transcribed as a complementary sequence of 3 nucleotides called a codon. A given codon specifies a particular amino acid. The genetic code is the set of rules that relate the base triplet sequence of DNA to the corresponding codons of RNA and the amino acids that specify. Transcription. During transcription, which occurs in the nucleus, the genetic information represented by the sequence of base triplets in DNA serves as a template for copying the information into complementary sequences of codons. 3 types of RNA are made from the DNA template. Messenger RNA, mRNA, directs the synthesis of a protein. Ribosomal RNA, rRNA, joins the ribosomal proteins to make ribosomes. Transfer RNA, tRNA, binds to an amino acid and holds it in place on a ribosome until it is incorporated into a protein during translation. One end of the transfer RNA carries a specific amino acid, and the opposite end consists of a triplet of nucleotides called an anticodon. By pairing between complementary bases, the tRNA anticodon attaches to the mRNA codon. Each of the more than 20 different types of tRNA binds to only one of the 20 different amino acids. The enzyme RNA polymerase catalyzes transcription of DNA. However, the enzyme must be instructed where to start the transcription process and where to end. Only one of the 2 DNA strands serves as a template for RNA synthesis. The segment of DNA where transcription begins, a special nucleotide sequence called a promoter, is located near the beginning of a gene. This is where RNA polymerase attaches to the DNA. During transcription, base pairs in a complementary manner. Bases pair in a complementary manner. The bases cytosine, guanine, and thymine in the DNA template pair with guanine, cytosine, and adenine, respectively, in the RNA strand. However, adenine in the DNA template pairs with uracil, not thymine in RNA. I'm going to repeat that. The enzyme RNA polymerase catalyzes transcription of DNA. However, the enzyme must be instructed where to start the transcription process and where to end it. Only one of the two DNA strands serves as a template for RNA synthesis. The segment of DNA where transcription begins, a special nucleotide sequence called a promoter, is located near the beginning of a gene. This is where RNA polymerase attaches to the DNA. During transcription, bases pair in a complementary manner. The bases cytosine, guanine, and thymine in the DNA template pair with guanine, cytosine, and adenine, respectively, in the RNA strand. However, adenine in the DNA template pairs with uracil, not thymine in RNA. Transcription of the DNA strand ends at another special nucleotide sequence called a terminator, which specifies the end of the gene. When RNA polymerase reaches the terminator, the enzyme detaches from the transcribed RNA molecule in the DNA strand. Not all parts of a gene actually code for parts of a protein. Regions within a gene called introns do not code for parts of proteins. They are located between regions called exons. They do code for segments of a protein. Immediately after transcription, the transcript includes information from both introns and exons and is called pre-mRNA. The introns are removed from pre-mRNA by small nuclear ribonucleoproteins. The snRNPs, or small nuclear ribonucleoproteins, are enzymes that cut out the introns and splice together the exons. The resulting product is a functional mRNA molecule that passes through a pore in the nuclear envelope to reach the cytoplasm where translation takes place. Although the human genome contains about 30,000 genes, there are probably 500,000 to 1 million human proteins. How can so many proteins be coded by so few genes? Part of the answer lies in alternative splicing of mRNA, a process in which the pre-mRNA transcribed from a gene is spliced in different ways to produce several different mRNAs. The different mRNAs are then translated into different proteins. In this way, one gene may code for 10 or more different proteins. In addition, chemical modifications are made to proteins after translation. For example, as proteins pass through the Golgi complex, such chemical alterations can produce 2 or more different proteins from a single translation. Translation. In the process of translation, the nucleotide sequence in an mRNA molecule specifies the amino acid sequence of a protein. Ribosomes in the cytoplasm carry out translation. The small subunit of a ribosome has a binding site for mRNA. The large subunit has 2 binding sites for tRNA molecules, a P site and an A site. The first tRNA molecule bearing its specific amino acid attaches to mRNA at the P site. The A site holds the next tRNA molecule bearing its amino acid. Translation occurs in the following way. An mRNA molecule binds to the small ribosomal subunit at the mRNA binding site. A special tRNA called initiator tRNA binds to the start codon, AUG, on mRNA, where translation begins. The tRNA anticodon, UAC, attaches to the mRNA codon, AUG, by pairing between the complementary bases. Besides being the start codon, AUG is also the codon for the amino acid methionine. Thus, methionine is always the first amino acid in a growing polypeptide. Next, the large ribosomal subunit attaches to the small ribosomal subunit mRNA complex, creating a functional ribosome. The initiator tRNA with its amino acid methionine fits into the P site of the ribosome. The anticodon of another tRNA with its attached amino acid pairs with the second mRNA codon at the A site of the ribosome. A component of the large ribosomal subunit catalyzes the formation of a peptide bond between methionine, which separates from its tRNA at the P site, and the amino acid carried by the tRNA at the A site. After peptide bond formation, the empty tRNA at the P site attaches from the ribosome, and the ribosome shifts the mRNA strand by one codon. The tRNA in the A site bearing the two peptide proteins shifts into the P site, allowing another tRNA with its amino acid to bind to a newly exposed codon at the A site. Steps 3 through 5 occur repeatedly, and the protein lengthens progressively. Protein synthesis ends when the ribosome reaches a stop codon at the A site, which causes the completed protein to detach from the final tRNA. When the tRNA vacates the A site, the ribosome splits into large and small subunits. Protein synthesis progresses at a rate of about 15 peptide bonds per second. As the ribosome moves along the mRNA, and before it completes synthesis of the whole protein, another ribosome may attach behind it and begin translation of the same mRNA strand. Several ribosomes attached to the same mRNA constitute a polyribosome. The simultaneous movement of several ribosomes along the same mRNA molecule permits the translation of one mRNA into several identical proteins at the same time. The clinical connection, recombinant DNA. Scientists have developed techniques for inserting genes from other organisms into a variety of host cells. Manipulating the cells in this way can cause the host organism to produce proteins it normally does not synthesize. Organisms so altered are called recombinants, and their DNA, a combination of DNA from different sources, is called recombinant DNA. When recombinant DNA functions properly, the host will synthesize the protein specified by the new gene it has acquired. The technology that has arisen from manipulation of genetic material is referred to as genetic engineering. The practical applications of recombinant DNA technology are enormous. Strains of recombinant bacteria now produce large quantities of many important therapeutic substances, including human growth hormone, required for normal growth and metabolism, insulin, a hormone that helps regulate blood glucose level and is used by diabetics, interferon, an antiviral and possibly anti-cancer substance, and erythropoietin, EPO, a hormone that stimulates production of red blood cells. Cellular division. The object is to discuss the stages, events, and significance of somatic and reproductive cell division. Describe the signals that induce somatic cell division. Most cells of the human body undergo cell division, the process by which cells reproduce themselves. The two types of cell division, somatic cell division and reproductive cell division, accomplish different goals from the organism. A somatic cell is any cell of the body other than a germ cell. A germ cell is a gamete, or any precursor cell destined to become a gamete. In somatic cell division, a cell undergoes a nuclear division called mitosis, and a cytoplasmic division called cytokinesis, to produce two identical cells, each with the same number and kind of chromosomes as the original cell. Somatic cell division replaces dead or injured cells and adds new ones during tissue growth. Reproductive cell division is the mechanism that produces gametes, the cells needed to form the new generation of sexually reproducing organisms. This process consists of a special two-step division called meiosis. in which the number of chromosomes in the nucleus is reduced by half. Again, this process consists of a special two-step division called meiosis, in which the number of chromosomes in the nucleus is reduced by half. Somatic cell division. The cell cycle in an orderly sequence of events by which a somatic cell duplicates its contents and divides in two. Human cells, such as those in the brain, stomach, and kidneys, contain 23 pairs of chromosomes for a total of 46. One member of each pair is inherited from each parent. The two chromosomes that make up each pair are called homologous chromosomes, or homologs. They contain similar genes arranged in the same, or almost the same, order. When examined under a light microscope, homologous chromosomes generally look very similar. The exception to the rule is one pair of chromosomes called the sex chromosomes, designated X and Y. In females, the homologous pair of sex chromosomes consists of two large X chromosomes. In males, the pair consists of an X and a much smaller Y chromosome. Because somatic cells contain two sets of chromosomes, they are called diploid cells. Once again, the exception of this rule is one pair of chromosomes called the sex chromosomes, designated X and Y. In females, the homologous pair of sex chromosomes consists of two large X chromosomes. In males, the pair consists of an X and a much smaller Y. Because somatic cells contain two sets of chromosomes, they are called diploid cells. When a cell reproduces, it must duplicate all its chromosomes to pass its genes to the next generation of cells. The cell cycle consists of two major periods: interphase, when a cell is not dividing, and the mitotic phase, when the cell is dividing. Interphase. During interphase, the cells replicate its DNA through a process that will be described shortly. It also produces additional organelle and systolic components in anticipation of cell division. Interphase is a state of high metabolic activity. It is during this time that the cell does most of its growing. Interphase consists of three phases: G1, S, and G2. The S stands for synthesis of DNA because the G phase are periods when there is no activity related to DNA duplication. They are thought of as gaps or interruptions in DNA duplication. The G1 phase is the interval phase between the mitotic phase and the S phase. During G1, the cell is metabolically active. It replicates most of its organelle and systolic components, but not its DNA. Replication of centrosomes also begins in the G1 phase. Virtually all the cellular activities described in this phase happen during G1. For a cell with a total cell cycle time of 24 hours, G1 lasts 8 to 10 hours. However, the duration of this phase is quite variable. It is very short in many embryonic cells or cancer cells. Cells that remain in G1 for a very long time, perhaps destined never to divide again, are said to be in the G0 phase. Most nerve cells are in the G0 phase. Once a cell enters the S phase, however, it is committed to go through cell division. The S phase, the interval between G1 and G2, lasts about 8 hours. During the S phase, DNA replication occurs. As a result, the two identical cells formed during cell division will have the same genetic material. The G2 phase is the interval between the S phase and the mitotic phase. It lasts 4 to 6 hours. During G2, cell growth continues. Enzymes and other proteins are synthesized in preparation for cell division, and replication of centrosomes is completed. When DNA replicates during the S phase, its helical structure partially uncoils and the two strands separate at the point where the hydrogen bond connects base pairs. Each exposed base of the old DNA strand then pairs with the complementary base of a newly synthesized nucleotide. A new DNA strand takes shape as chemical bonds from between neighboring nucleotides. a new DNA strand takes shape as chemical bonds form between neighboring nucleotides. The uncoiling and complementary base pairing continues until each of the two original DNA strands is joined with a newly formed complementary DNA strand. The original DNA molecule has become two identical DNA molecules. A microscopic view of a cell during interphase shows a clearly defined nuclear envelope, a nucleolus, and a tangled mass of chromatin. Once a cell completes its activity during the G1S and G2 phase of interphase, the mitotic phase begins. Mitotic phase. The mitotic, or M phase, of the cell cycle consists of a nuclear division, mitosis, and a cytoplasmic division, cytokinesis, to form two identical cells. The events that occur during mitosis and cytokinesis are plainly visible under a microscope because chromatin condenses into discrete chromosomes. Nuclear division, mitosis. Mitosis, as noted earlier, is the distribution of two sets of chromosomes into two separate nuclei. The process results in the exact partitioning of genetic information. For convenience, biologists divide the process into four stages: prophase, metaphase, anaphase, and telophase. However, mitosis is a continuous process. One stage merges directly into the next. Prophase. During early prophase, the chromatin fibers condense and shorten into chromosomes that are visible under the light microscope. The condensation process may prevent entangling of the long DNA strands as they move during mitosis. Because longitudinal DNA replication took place during the S phase of interphase, each prophase chromosome consists of a pair of identical strands called chromatids. A constricted region called a centromere holds the chromatid pair together. At the outside of each centromere is a protein complex known as kinetochore. Later in prophase, tubulins in the pericentriolar material of the centrosome start to form the mitotic spindle, a football-shaped assembly of microtubules that attach to the kinetochore. As the microtubules lengthen, they push the centrosomes to the poles, the ends, of the cell so that the spindle extends from pole to pole. The mitotic spindle is responsible for the separation of chromatids to opposite poles of the cell. Then the nucleolus disappears and the nuclear envelope breaks down. Metaphase. During metaphase, the microtubules of the mitotic spindle align in the centromeres of the chromatid pairs at the exact center of the mitotic spindle. Once again, metaphase. During metaphase, the microtubules of the mitotic spindle align the centromeres of the chromatid pairs at the exact center of the mitotic spindle. This midpoint region is called the metaphase plate. Anaphase. During anaphase, the centromeres split, separating the two members of each chromatid pair, which moves towards opposite poles of the cell. Once separated, the chromatids are termed chromosomes. As the chromosomes are pulled by the microtubules of the mitotic spindle during anaphase, they appear V-shaped because the centromeres lead the way, dragging the trailing arms of the chromosomes towards the poles. Telophase, the final stage of mitosis. Telophase begins after chromosomal movement stops. The identical set of chromosomes, now at the opposite pole of the cell, uncoil and revert to thread-like chromatin forms. A nuclear envelope forms around each chromatin mass. Nucleoli reappear in the identical nuclei, and the mitotic spindle breaks up. Cytoplasmic division, cytokinesis. As noted earlier, division of cells, cytoplasm, and organelle into two identical cells is called cytokinesis. This process usually begins in the late anaphase with the formation of a cleavage furrow, a slight indentation of the plasma membrane, and is completed after telophase. The cleavage furrow usually appears midway between the centrosomes and extends around the periphery of the cell. Actin microfilaments that lie just inside the plasma membrane form a contractile ring that pulls the plasma membrane progressively inward. The ring constricts the center of the cell like tightening a belt around the waist and ultimately pinches it in two. Because the plane of the cleavage furrow is always perpendicular to the mitotic spindle, the two sets of chromosomes end up in separate cells. When cytokinesis is complete, interphase begins. The sequence of events can be summarized as G1 yields S phase, which yields G2 phase, which yields mitosis, which yields cytokinesis. The clinical connection, mitotic spindle in cancer. One of the distinguishing features of cancer cells is uncontrolled division. The mass of cells resulting from this division is called neoplasm or tumor. One of the ways to treat cancer is by chemotherapy, the use of anti-cancer drugs. Some of these drugs stop cell division by inhibiting the formation of the mitotic spindle. Unfortunately, these types of anti-cancer drugs also kill all types of rapidly dividing cells in the body, causing side effects such as nausea, diarrhea, hair loss, fatigue, and decreased resistance to disease. Control of cell destiny. A cell has three possible destinies: to remain alive and functioning without dividing, to grow and divide, or to die. Homeostasis is maintained when there is a balance between cell proliferation and cell death. The signals that tell a cell when to exist in the G0 phase, when to divide, and when to die have been the subjects of intense and fruitful research in recent years. Within a cell, there are enzymes called silin-dependent protein kinases that can transfer a phosphate group from ATP to a protein to activate the protein. Other enzymes can remove the phosphate group from the protein to deactivate it. The activation and deactivation of CDKSs, or cyclin-dependent protein kinases, at the appropriate time is crucial in the initiation and regulation of DNA replication, mitosis, and cytokinesis. Switching the CDKS on and off is the responsibility of cellular proteins called cyclins, so named because their levels rise and fall during the cell cycle. The joining of a specific cyclin and CDK molecule trigger various events that control cell division. The activation of specific cyclin CDK complexes is responsible for progression of a cell from G1 to S to G2 to mitosis in a specific order. If any step in a sequence is delayed, all subsequent steps are delayed in order to maintain the normal sequence. The levels of cyclins in the cell are very important to determining the timing and sequence of events in cell division. For example, the level of the cyclin that helps drive a cell from G2 to mitosis rises throughout the G1, S, and G2 phases and into mitosis. The high level triggers mitosis, but towards the end of mitosis, the level declines rapidly and mitosis begins. Destruction of this cyclin, as well as others in the cell, is by proteasomes. Cellular death is also regulated. Throughout the lifetime of an organism, certain cells undergo apoptosis, an orderly, genetically programmed death. In apoptosis, a triggering agent from either side or inside the cell causes cell suicide. Genes to produce enzymes that damage the cell in several ways, including disruption of its cytoskeleton and nucleus. As a result, the cell shrinks and pulls away from neighboring cells, although the plasma membrane remains intact. The DNA within the nucleus fragments and the cytoplasm shrinks. Phagocytes in the vicinity then digest the dying cell. The function of phagocytes involves a receptor protein in the plasma membrane of the phagocyte. that binds to a lipid in the plasma membrane of the suicidal cell. Apoptosis removes unneeded cells during fetal development, such as the webbing between the fingers, webbing between the digits. It continues to occur after birth to regulate the number of cells in a tissue and eliminate potentially dangerous cells, such as cancer cells. Apoptosis is a normal type of cell death. In contrast, necrosis is a pathological type of cell death that results from tissue injury. In necrosis, many adjacent cells swell, burst, and spill their cytoplasm into the interstitial fluid. The cellular debris usually stimulates an inflammatory response by the immune system, a process that does not occur in apoptosis. The events of the somatic cell cycle revisited. Interphase, period between cell division, chromosomes, not visible under light microscope. G1 phase, metabolically active cells duplicate organelles and systolic components. Replication of chromosomes begins. Cells that remain in the G1 phase for a very long time and possibly never divide again are said to be in the G0 phase. Replication of DNA in centromeres, S phase. G2 phase, cell growth enzyme in protein synthesis continues. Replication of centrosomes complete. Mitotic phase, parent cell produces identical cells with identical chromosomes, chromosomes visible under light microscope. Mitosis, nuclear division, distribution of two sets of chromosomes into separate nuclei. Prophase, chromatin fibers condense into paired chromatids, nucleolus and nucleus. Prophase, chromatin fibers condense into paired chromatids. Nucleolus and nuclear envelope disappear. Each centrosome moves to an opposite pole of the cell. Metaphase, centromeres of chromatid pairs line up at the metaphase plate. Anaphase, centromeres split, identical sets of chromosomes move to opposite poles of the cell. Telophase, nuclear envelopes and nucleoli reappear. Chromosomes resume chromatin form. Mitotic spindle disappears. Cytokinesis, cytoplasmic division. Contractile rings forms cleavage furrow around the center of a cell, dividing cytoplasm into separate and equal portions. Reproductive cell division. In the process called sexual reproduction, each new organism is the result of the union of two different gametes, one produced by each parent. If gametes had the same number of chromosomes as somatic cells, the number of chromosomes would double at fertilization. The reproduction cell division that occurs in the gonads, ovaries, and testes is meiosis. It produces gametes in which the number of chromosomes is reduced by half. As a result, gametes contain a single set of 23 chromosomes and thus are haploid cells. Fertilization restores the diploid number of chromosomes. Once again, reproductive cell division is the process called sexual reproduction. Each new organism is the result of the union of two different gametes. Fertilization, one produced by each parent. If gametes had the same number of chromosomes as somatic cells, the number of chromosomes would double at fertilization. Meiosis, which means to lessen. Meiosis, the reproductive cell division that occurs in the gonads, ovaries, and testes, produces gametes in which the number of chromosomes is reduced by half. As a result, gametes contain a single set of 23 chromosomes and thus are haploid. HAPL, hapl, meaning single. Fertilization restores the diploid number of chromosomes. Meiosis. Unlike mitosis, which is complete after a single round, meiosis occurs in two successive stages, meiosis I and meiosis II. Continued. Meiosis. Unlike mitosis, which is the complete, meiosis. Unlike mitosis, which is complete after a single round, meiosis occurs in two successive stages, meiosis I and meiosis II. During the interphase that precedes meiosis I, the chromosomes of the diploid cell start to replicate. As a result of replication, each chromosome consists of two sister, genetically identical, chromatids, which are attached at the centromeres. This replication of chromosomes is similar to the one that precedes mitosis in somatic cell division. Meiosis I. Meiosis I, which begins once chromosomal replication is complete, consists of four phases: prophase I, metaphase I, anaphase I, and telophase I. Prophase I is an extended phase in which the chromosomes shorten and thicken. The nuclear envelope and nucleoli disappear and the mitotic spindle forms. Two events that are not seen in mitotic prophase occur during prophase I of meiosis. First, the two sister chromatids of each pair of homologous chromosomes pair off. An event called synapsis. The resulting four chromatids form a structure called a tetrad. Second, parts of the chromatids of two homologous chromosomes may be exchanged with one another. Such an exchange between parts of non-sister, genetically different chromatids is termed crossover. I'd like to repeat that. Meiosis I. Meiosis I, which begins once chromosomal replication is complete, consists of four phases: prophase I, metaphase I, anaphase I, and telophase I. Prophase I is an extended phase in which the chromosomes shorten and thicken. The nuclear envelope and nuclei disappear and the mitotic spindle forms. Two events that are not seen in mitotic prophase occur during prophase I of meiosis. First, the two sister chromatids of each pair of homologous chromosomes pair off, an event called synapsis. The resulting four chromatids form a structure called a tetrad. Second, parts of the chromatids of two homologous chromosomes may be exchanged with one another. Such an exchange between parts of non-sister chromatids is termed crossing over. This process, among others, permits an exchange of genes between chromatids of homologous chromosomes. Due to crossing over, the resulting cells are genetically unlike each other and genetically unlike the starting cell that produced them. Crossing over results in genetic recombination, that is, the formation of new combinations of genes, and accounts for part of the great genetic variation among humans and other organisms that form gametes via meiosis. In metaphase I, the tetrads formed by the homologous pairs of chromosomes line up along the metaphase plate of the cell, with homologous chromosomes side by side. During anaphase I, the members of each homologous pair of chromosomes separate as they pull to opposite sides of the cell by the microtubules attached to the centromeres. The paired chromatids held by a centromere remain together. Recall that during mitotic anaphase, the centromeres split and the sister chromatids separate. Telophase I and cytokinesis of meiosis are similar to telophase and cytokinesis of mitosis. The net effect of meiosis I is that each resulting cell contains the haploid number of chromosomes because it contains only one member of each pair of the homologous chromosomes present in the starting cell. Meiosis II. The second stage of meiosis, meiosis II, also consists of four phases: prophase II, metaphase II, anaphase II, and telophase II. These phases are similar to those that occur during the mitosis. The centromeres split and the sister chromatids separate and move towards opposite poles of the cell. In summary, meiosis I begins with a diploid starting cell and ends with 2 cells, each with a haploid number of chromosomes. During meiosis II, each of the 2 haploid cells formed during meiosis I divides. The net result is 4 haploid gametes that are genetically different from the original diploid starting cell.